Yep. So we're just moving on to the next session, which is going to be progress in the treatment landscape, and who better to kick us off than John Bridgewater. Thanks.
Thank you very much, everybody. Very tough act to follow, as ever. So this is about clinical trials and a bit more about the ABC-10 trial, SAFIR ABC-10 trial. Some people found out a bit about it yesterday. But if anything, this just tells us, makes the point that Andrea was trying to make earlier, that so much can be done, and actually remarkably little is being done. And that unfairness, that disparity, I personally think is pretty scandalous. But I think that's the single piece of information that perhaps should be the most important. But in the meantime, I'll pass the time by telling you about the available clinical trials and what we're doing in SAFIR ABC-10. And once again, thank you, Helen, Heinz-Josef, Rachel, the committee, and so on for asking me. It's, as ever, a great pleasure. Short introduction about trials. So clinical trials or studies are generally broken down into three groups. So the phase one is what is commonly known as the first-in-man studies. So these are guinea pig things, never previously given to human beings, but extremely promising for a number of reasons, and there can be lots of those. And this is generally given to people to see whether it's safe or not. Most things are reasonably safe, but some things are not safe, and there's a lot of history of things going horribly wrong. But as a rule, they are generally extremely safe. But these trials are designed to make sure they are safe, and to find the dose for the next bit, which is the phase two, which is something that works out how well it works, whether it works or not. And these can be just everybody getting this stuff, or they can be randomized, which is where half the people get it, or a proportion of people get it and a proportion of people don't. And then once you've established, actually, this can do something, this may work, then you do a phase 3 trial where you compare it against what is done as the standard. That's what we call the standard of care. And there's various ways of doing that. The size of these studies increases as you go through them. And that needs to happen because the statistics need to be done to make them supportive of the outcome. The thing that applies to most people are phase 3 studies. These are big studies with 2 or more arms, where the outcome, in other words, the measurement of whether it's worked or not, is in terms of is measured in terms of how long people live, the benefit, or indeed the quality of life. And over the last, golly, too long, 20 plus years, myself and at least several people in this room, Juan, good afternoon, have demonstrated the role of chemotherapy in cholangiocarcinoma and biliary tract cancer as a rule. And that's as adjuvant, so you heard about that this morning from John Primrose. That's capecitabine as adjuvant. The first-line treatment, the ABC-2 study, which we published in 2010, and then the second-line study. So the treatment. So the treatment you give for people with advanced disease at the beginning, and then when that doesn't work, FOLFOX. And that the latest advance on that is the first-line treatment. It's cisplatin and gemcitabine, but with the addition of durvalumab or pembrolizumab. In the UK, we can only use durvalumab, but that has become the complete standard of choice. But really, it's the targeted therapies that have generated interest and excitement, perhaps only over the last 10 years. And I've given this talk once or twice, and this talk changes every few months because the advances are so significant. And this was brought about originally from a thing called the Cancer Genome Atlas. So this is a pile of international money that went into sequencing these cancers. And sequencing in cancer is a fairly standard thing to do today, but 15 years ago it was really quite a big deal and immensely expensive. But almost 500 patients were sequenced with cholangiocarcinoma and were sequenced. And it transpired that for some reason, and we still don't know why, cholangiocarcinoma has more than the average number of molecular alterations, so genomic alterations in the DNA that drive it, and these are distributed in particular patterns through the various types of cholangiocarcinoma you can get, and that was really where it started, something like 10 to 15 years ago. And this has led to a development of drugs to treat those targets. And these are the latest set of ESMO guidelines. There's a live update on it of the system. And you can divide this up into several chunks. So first of all, this is the post-surgery situation where patients are currently given capecitabine, but hopefully by October this year we may be using something else. The Actica trial will report in October of this year. We hope. It's a late-breaking abstract, in fact. So we may not actually have the numbers, but perhaps we will. The other common situation, the much more common situation in which targeted therapies are given are, of course, in advanced disease where the cancer has spread, and there are now multiple different targets for these the patients who have these particular alterations. So the IDH1 mutation, FGFR2 fusions, and the mismatch repair, so the things in blue are things that are reimbursed in the UK. This whole guideline also describes alterations that can be treated in the BRAF gene and in the HER2 gene, and the drugs have been approved in the US for both of these indications. So these are what we would consider to be the current usual suspects in terms of actionable alterations. So and it is ever so important to know whether you have one of these. So first of all, so what are the studies available at the moment? So I'll just run through the current studies available at the moment and finish off with a description of ABC-10, which I think summarizes to a certain extent what I've spoken about just now. So this is the the list of the current studies that I have, by no means exhaustive, is that I'm going to describe is up on the left here. And this first one is the Artemide study. There's a poster next door about it. And essentially, it's the next step along from the BILCAP study, where patients with cancer that's been resected are given either the standard chemotherapy or the standard chemotherapy with this drug, rilvegostomig. John Primrose couldn't pronounce it. It's pronounced rilvegostomig, and it's what we call it's the new generation of antibodies. So just here, this is an antibody, and antibodies commonly have two arms, and generally speaking, they're identical arms. They hit the same thing. In the past, maybe 5 years, this thing called the bispecific, in which one arm targets one thing and the other arm targets the other thing, have really started to hit the shelves. And this targets the standard thing that an immunotherapy drug targets, which is PD-L1, and the other arm of it targets another type of immunotherapy target called TIGIT. And so this is a bispecific PD-L1 TIGIT, and the idea is that it will improve your own immune system's attack against any residual cancer. And the preliminary data were very promising, and so this is recruiting really well, actually, much better than originally anticipated. And in fact, this study will now finish in probably close towards the end of this year, having recruited more than 600 patients. So that's really promising, and will tell us whether immunotherapy works as adjuvant, so after surgery. What are the other studies? Well, the next two I'm going to describe look at the target called HER2. So HER2, here we go. HER2, so this is the first one. This is the zanidatamab study. But first of all, HER2 is a target in several cancers, primarily breast cancer. So a lot of very good drugs have been made to target HER2 because of breast cancer. Because breast cancer has the numbers, has the in some ways popular backing, if you like, to support all this development. This drug is a variation on the bispecific zanidatamab. So normally one of the arms of your antibody will the arms of your antibody will be the same thing, targeting this thing on the cell surface called HER2. Zanidatamab targets two different bits of the HER2 receptor, and so instead of just going in there and targeting the one thing, it forms chains, it forms carpets on the surface of the target cell, and consequently, this is thought to be why it could be more effective. And so here is a study that is a randomized phase 3 study. So it compares the standard, which is the cisplatin gemcitabine and durvalumab standard, and it compares it to that standard plus zanidatamab, and that's up and running at the moment. Very similar, but you have to have the HER2. You have to have the HER2. That's probably about 1 in 6, 1 in 7 patients with cholangiocarcinoma. A very, very similar design is the AstraZeneca study, where patients, where this is the drug that's being investigated to a large part, in HER2. So in HER2, or trastuzumab deruxtecan, sorry about all these names, is a standard HER2 binder, so different to the last one I described, different to the Zani, but it's got chemotherapy attached to the end of it. Consequently, that chemotherapy is delivered to the cancer cell at concentrations several thousand-fold higher than if you just simply had chemotherapy injected to you. The chemotherapy here is described as the payload, and this bomb attacks the HER2 expressing cell directly. And here's the study design, looking at an interesting combination of the standard treatment here versus Enhertu by itself, versus Enhertu and rilvegostomig, which I've just described. So that's up and running at the moment, and also available. I'm just going to run to the second line study here first before going into ABC-10. And this is a second-line study for people with FGFR2 alterations. So FGFR2 is one of the targets that we find, and we have some really good drugs looking at FGFR2 in the the cartoon is down here really. So your standard FGFR2 binds to your FGFR2 cell, your FGFR2 driven cancer, and but eventually they become at some stage they normally become resistant to that, and this is a drug that's thought to have be effective in those people in whom it's become resistant. And so this is a study that's currently running. In fact, we're using the 10 milligram dose here, and the 10 milligrams dose here, and it's a randomized study. So 2 people get the stuff, and 1 person gets chemotherapy. There's a lot of pressure for them to make it what we call a crossover study, so that when the chemotherapy doesn't work, you then go on and have potentially these tablets, potentially the good stuff, because all sorts of reasons, but there's a lot of pressure for them to do that, and in fact I hope that's going to happen. So that's the tinningotinib study. So not an exhaustive list, but the ones I think that are the most interesting. But I'll spend the last 5 minutes now talking about this. I mentioned this yesterday, and this is the lead study in the UK and France at the moment, the lead academic study, and it took us ages to get it started, because we had to get academic charity money from 3 countries, Belgium, France, and the UK. But started we have, and this is probably the first report anywhere of how well we're doing. So, there's 2 main bits to this study. It's about the testing up at the top, and then the treatment down here at the bottom. And the testing is 2 tests simultaneously. It's a tissue test, so a test of the biopsy or the material, and it's a liquid biopsy. And I think you've heard over the last day and a half how important it is really to get the best test you can. And I think this study actually is going to tell us a great deal about what the best study is, what the best testing is. And in many ways, I think this bit, the testing bit, is going to be more interesting than the treatment bit. We reckoned about 1 in 4 patients would pass the test in terms of finding something we could hit, one of these targets and the drugs to target it on the left here, and then people would be randomized so that 2 out of 3 people would get the targeted therapy. 1 out of 3 people would continue on the standard arm until that didn't work anymore, and then they'd move on to the targeted therapy. So it was really a question of when you got the stuff rather than whether you got the stuff at all. So the crossover design permits that. So this is the basic design for the SAFIR ABC-10 study. And these are the drugs that we use. I can go into that in due course. And this is a recruitment graph, and this is now about 2 months, 6 weeks out of date. And so there's 2 bits of it. So there's the people we did the test on, and then the people who've been randomized into the study. And this number, so the green line is what we actually are doing, and the dotted line is what we're supposed to be doing. And we're now up to, this is 6 weeks out of date, we're now up to well over 200. And the study is really recruiting remarkably well. And instead of finishing somewhere sometime in 2027, we're probably going to finish in the middle of next year, so a year ahead of time. So this is a huge tribute to everybody out there who's contributing to this study. You know, if anything else, it tells us how to run these studies, how to make it possible for patients. So it's going well. It's going well. We're running neck and neck with the French at the moment. No contest. These are the targets that have been found. So we're finding more targets than we expected. And that's got to do with, I'll go into it at some stage if you want, but it's got to do with using 2 tests and the best tests. So about two-thirds, this is in fact 38% of people have an actionable alteration. So that's quite a lot. I reckon that's quite a lot. That's more than we thought. And for some of these, for some of these people here, it's a real game changer. Slightly more of some things than the others, but this is still relatively early, and this is still only about 90 people. But we don't really expect this to change. We have a big meeting with the French to go through this information, and it's the most well-attended meeting that everybody goes, because the discussions about each one of these, how you get to the answer, what's the best way of treating it, are some of the most interesting discussions I've ever had. And these are the people being treated at the moment. Thank goodness, technical failure is very, very limited. No target 55. We'd sort of expected that, but oh yes, and this is a sort of timeline of what's going on. So screened, alteration found. Some people don't get randomized for one reason or another, and so that's really going pretty well. The other thing that we didn't anticipate, and is hugely exciting as far as I'm concerned, are the extras, are the spin-offs. So that's the core study on the left there. But in addition to that, we found some other targets, and other pharma have contacted us saying, look, you're doing all this testing. Are you by any chance treating this particular alteration? And so there are now several extras being considered here. So for instance, IDH2, for which Servier have a drug. We've had conversations at this meeting with Servier about it. Already we have found 6 people with IDH2 alterations, much more than originally intended. And so there's a plan now to develop a new extra study consequent on that. We were approached by a biotech from the States with a BAP1 drug. And so there's an application gone through to Cancer Research UK with that on it. This is one of, in fact, 2 RAS. So RAS is a bad guy in a lot of these cancers, constitutes about 20% of all of our patients. This is one of, in fact, 2 applications going forward for RAS drugs. There's a radiation component, and then there's a second line immunotherapy treatment study called SAFIR starting up. So it's what these big studies, these big platform studies are supposed to do. They're supposed to stimulate interest, stimulate testing, stimulate other studies to follow it. And I'm incredibly grateful to my colleagues for helping us develop this and move it on in this way. So that's what's going on at the moment. As you can see, very fluid, everything to play for as it runs currently. In summary, it's a really active research environment out there. When Juan and I started, we were the only study, pretty much. Maybe a handful of other little studies, but we were the only study. Now the whole thing is awash, and that must be a good thing. We have found 36% of patients in ABC-10 have actionable alterations within the study. There are other actionable alterations outside of the study. This is worth doing it all again for just by itself, really. And the platform, the whole sort of process of doing the best test, seeing what your patient's got in terms of something you can treat, that hopefully will turn out to be the most valuable part of this. So all the people who pay for us, thanks very much indeed.